Hey guys, this is Mr. Bennett. This is our third biochemistry video. Today we're going to be talking about carbohydrates and lipids found on pages 51 to 60 in the textbook. Now I want to do a little bit of review before we get started. So the first thing I want to review is the concept of a monomer and a polymer. Remember that monomers are the single pieces, the single pieces of the large molecules in our body. So mono meaning single. And if we take these monomers and we bond them together, we can form a polymer. So polymer is, generally speaking, 3 or more monomers combined. It could be 2 technically, but we bond them together and then they serve a purpose within the body. Polymers may be really tiny, could be again just 2 little monomers, or they could be hundreds of monomers long. So there are 4 categories of polymers or macromolecules that we're going to talk about. We talked about 2 a couple days ago. The first one would be proteins. Now, proteins are really long chains of a monomer called an amino acid. So in this case, the amino acid is the monomer, and then the polymer is called a polypeptide. If this polypeptide has a function or does a job within the cell, then it becomes a protein. There's really 2 different categories of proteins, but they're all really tools. I mean, if we were to really look at the general use of a protein, it's a tool within the cell. But the 2 big ones are enzymes, it's a big category of protein, and structural. So basically they form structures within the cell. Okay, so those are the 2 uses. They're monomers of amino acid, polymers of polypeptide. And one other thing I want to remind you of about proteins is that it's all about their shape. Because they are tools, their shape is huge. Their shape is tied to their function. Okay, so a protein is made up of lots of amino acids all bonded together using peptide bonds. Okay, a chain like this, and then that chain will fold up into a 3-dimensional shape. That shape is directly tied to its function. Now the second category of macromolecules that we've already talked about are nucleic acids. Nucleic acids, their job is to store and transport information within the cell. Their monomer is a nucleotide. Okay, there are 5 different nucleotides that they can use, and again the polymer would be a nucleic acid. The 2 big examples of these guys would be DNA and RNA. Okay, DNA stores the information, genetic information within the cell. RNA, on the other hand, can store and transport it. Okay, so very similar to DNA, proteins, in that we're chaining together a monomer, but notice it's a different type of monomer, and therefore we get a different function. Okay, so we're going to see this chart twice. Right now in your notes you see a chart that has only 2 rows. The 2 rows I want you to do right now are proteins and nucleic acids. So based on what you know and based on what we just reviewed, I'd like you to fill in monomer, polymer, use an example for proteins and nucleic acids. Just use the 2 rows that you have. Okay, so I want you to pause the video, fill it in on your own, and then when you unpause it, I'll show you can check your answers. Okay, so pause it. Alright, hopefully you paused it. Let's fill these in real quick. So the protein monomer is an amino acid, right? And then that's the monomer. The polymer would be a polypeptide. The use, there's really 2 different uses. It can be an enzyme. Enzymes are all about catalyzing reactions, so catalyst. Okay, or it could be structural. I mentioned that. And an example, DNA polymerase is one enzyme that we talked about in our previous video. It is an enzyme that basically catalyzes the formation, the production of the polymer of DNA. So it helps make DNA. Remember, just a little side note, all enzymes, the last 3 letters of their name will be ACE. That's our tip that, or our flag that it's an enzyme. Nucleic acids, let's see what we got here. So a nucleic acid, the monomer would be a nucleotide, right? Nucleotide. And then the polymer would be, there's no real fancy name for it, so we would just say DNA or RNA. Okay. Then as far as use goes, we said to store or transport information. That's really the use of DNA or RNA. So, and I'm going to abbreviate it as info. Okay, and an example again would be DNA or RNA. Sorry, my handwriting's getting bad there at the bottom. I don't know why it always messes up down here. Anyhow, Moving on, so we've talked about two categories of macromolecules. The third category would be carbohydrates. Carbohydrates are used for energy storage as well as to form structural materials. So two different uses here. The monomer for a carbohydrate is known as a monosaccharide. So I actually like this one a lot because we can see right off the bat, it's kind of a hint here that it is the monomer. It's a single, and then in this case, saccharide, think of it as meaning sugar. So it's a single sugar or simple sugar. The main example of a simple sugar that I want you to know is this guy here, glucose. Okay, we're going to use him all year long. He is a simple sugar. Notice that all of the monomers here, we have three different monomers, three different carbohydrates or monosaccharides, glucose, fructose, and galactose. All three have ring shapes, okay, ring structure, just like nucleotides, only nucleotides had a nitrogen. These guys do not have a nitrogen. They're only made up of carbon, hydrogen, and oxygen, so no nitrogen. Macromolecule, if we take these monomers, we combine them together. First of all, we can make a disaccharide, or two monosaccharides joined together. Again, the word, use these prefixes to help you. Di means two, so it's two sugars put together. The most classic example that we know, that we see every day, or maybe experience fairly often, would be table sugar, also known as sucrose. It's a combination of glucose and fructose bonded together. Okay. If we take three or more monosaccharides and we bond those together, that's going to give us a polysaccharide. So poly means three or more. And we call these guys complex sugars. Okay, there's really, I'm going to show you four different examples. There's three you need to know. So the first one you need to know is glycogen. Glycogen is used for energy storage in animals. In fact, what we do is we take glucose molecules, the guy on the previous slide, and we chain them together to make glycogen. Okay, you get glucose from your food. You need to store it away in your cells when you don't need it, and so it gets chained together in a molecule called glycogen. Plants, on the other hand, when they store their energy in the form of carbohydrates, they store it as starch. Okay. The weird thing about this, though, is that starch is made up of the same monomer. They take glucose, they bond it together, and they make a slightly different molecule than we do, just because the bond is a little bit different. The third category, or third example of polysaccharide that I want you to know, is cellulose. Cellulose is structural. Notice these two guys up here were energy related. This is used for structure. It helps make the cell wall in the plant for strength and rigidity of the cell. And another weird thing is that cellulose, just like glycogen and starch, is made up of glucose. So again, slight difference in bonding. All of a sudden, the polymer is good for structural purposes, making cell walls, compared to the energy storage. The fourth one I was going to show you, again, I'm just telling you about it, you don't need to know this one, is chitin. Chitin is the structure found in the cell walls of fungi and exoskeleton of insects. Another weird thing, glucose is involved in the formation of chitin as well. So same monomer creates four different polymers, all with different purposes. Now, that's carbohydrates real quick. Now we're moving on to the fourth and final category of macromolecules, the lipids. Big thing about the lipids, the thing that sets them aside from all other macromolecules, is that they are nonpolar, which means that they don't dissolve in water. Okay, the major purpose of lipids is to store energy, but they can also be used in some cases to make cell membranes. The monomer of a lipid is a fatty acid. Fatty acids are unbranched carbon chains, and they have a hydrophobic end and a hydrophilic end. So what does that really mean? Well, hydro means water, okay, so in both names it means water, but the ending is what's important here. Phobic means to fear, so think of like phobia. If I'm arachnophobic, I'm afraid of spiders. In this case, the molecule does not like to interact with water, so therefore we think of it as being water, afraid of water. Philic, on the other hand, means to love, so there's an end of the molecule that loves water. Okay, and again, lipids are all about storing lots and lots of energy. Here's an example of that monomer. So this would be two different examples of fatty acids, but in both cases, notice there's this big chain of carbons here. Okay, both of these guys are the hydrophobic ends. Okay, they don't like water, they don't like to interact with them. The hydrophilic end, on the other hand, is right up here. Okay, these purple ends right here, those are the hydrophilic ends. They love water, but notice they're very, very tiny. So as a whole, fatty acids do not like water. Most of the molecule is hydrophobic. Now, when we talk about lipids, there's really four types of lipids that we need to know. Two of them are big, meaning important. Two of them are just, you need to be familiar with them. So the first big one, the first important one, is known as a triglyceride. Tri, again, I can't emphasize this enough. Get used to prefixes, learn what they mean. So in this case, tri means 3. So that means that we have 3 of the monomers, the fatty acids, that are joined to a molecule that we call glycerol. So let me show you an example. All right, first of all, triglycerides are known as fats and oils. That's like the generic term. Fats would be solid at room temp, whereas oils are liquid at room temp. It's kind of the big difference here. Otherwise, molecularly, they look very similar. And what every triglyceride is made of is, first of all, a glycerol. Okay, notice there's these OHs right here. These OHs, think of them as connectors. The fatty acid can come in here and it can connect at that spot. So if I redraw this, and here is my glycerol molecule, which I think of as like a capital E, then my fatty acid molecule can come in here and form a bond. And another one can come in and form a bond right here, and another one can come in and form a bond right here. So that I have 1, 2, 3 fatty acid molecules that are bonded to that glycerol, hence triglyceride. This is another way of drawing it. We're just showing the individual atoms as spheres, but still you have 1, 2, 3 fatty acid molecules bonded to a glycerol. And then here is a line drawing of the same thing. So triglycerides. Second category would be the phospholipids. Very, very similar to a triglyceride with one exception. Instead of having 3 fatty acids bonded, you have 2. Okay, still have the glycerol, but in the place of that third fatty acid, we now have a phosphate. So kind of some complicated drawings here, but let's just explain it. First of all, phospholipids, the number one reason why they're important, the reason why we need to know these guys, is because they make up cell membranes. So next unit, unit 3, when we start talking about cells, these guys are really important because they're like one of the most important organelles. They make up it, okay? Now, let's talk about what a phospholipid really looks like, okay? If we go back to the idea that you have a glycerol molecule, we bond 2 fatty acid molecules, because phospholipids have 2 of them. They get bonded on right here and right here, okay? And then at the third spot, we add on a phosphate group instead of that third fatty acid that a triglyceride would have. Now, what you gotta know is that phospholipids, okay, they are just like the fatty acid in that there's a part of them that loves water and there's a part that hates it. We know that these fatty acids hate water. So this end right here, okay, this is phobic, meaning it hates water. Whereas this guy here is philic. It loves water, the phosphate. So what happens is, it turns out phobic and philic, philic, there we go. Not Phyllis. Phyllis is a person. Ha. Anyways, moving on. So the philic end and the phobic end, they're afraid of each other. So they will split apart. This guy will kind of rotate around this way, and you end up with a phosphate group. I'm just going to redraw it up here. And you still got your glycerol, okay, and attached to it you still have your 2 fatty acid molecules. This can be simplified into one of these guys where you have the phosphate and the 2 tails. This end loves water, this end doesn't, okay? So that's a phospholipid. Other 2 categories of lipids, you have waxes. Waxes are all about waterproofing. They form protective layers. For instance, a leaf that's on a plant that's in a really hot area where it's worried about losing water, it may coat its leaf with waxes to make them waterproof so that the water is trapped within the leaf and doesn't evaporate as fast. It's a protective layer. The fourth and final category of lipids is known as a steroid. The big thing about steroids is that they don't actually contain the monomer. They don't contain fatty acids. Instead, they're made up of 4 fused carbon rings, something like this. 2 examples. Cholesterol is the one you need to know, but also other steroids, things like testosterone. Notice there's 4 rings of carbon. They're all fused together. And again, the big thing about steroids is that they do not contain the monomer. So the question you might ask is, why are steroids considered lipids? Well, the big thing, as I said at the beginning of the lipid section, is that they are non-polar. They do not mix with water. So since they're made by us, like meaning our living things make them, and they're macromolecules, and they don't mix with water, we put them in here with lipids. Alright, so I mentioned that we'd see this again. What I'd like you to do right now is, based on the information that you just learned about carbohydrates, go back into your notes and fill in the 4 boxes. Monomer polymer use example for carbs, and then do it also for lipids. Okay, So pause it, and then you can start it up and check your answers. Okay, so pause now. All right, hopefully you paused it. Hopefully you're ready to check your answers. So for carbohydrates, the monomer is a monosaccharide. Okay, I'm going to abbreviate it as monosac. The polymer would be a polysaccharide. I'll abbreviate that as polysac. Use is for storing energy. And hopefully get the second use as well, and that would be structural. For example, cellulose making up the cell wall of plants. Examples, if we're talking monosaccharide, like monomer, could be glucose. If we're talking disaccharide, it would be sucrose. And then remember there's 3 polysaccharides that you need to know. They are glycogen, starch, and cellulose. Okay. Now moving on to lipids, the monomer is a fatty acid. The polymer, there's really 4 of them, so let's name them off. Triglyceride is 1. Then you have the phospholipid. Okay, so if I were to pick of the 4, the 2 that you need to know most about are triglycerides and phospholipids. You need to be familiar with waxes. Waxes. And number 4, steroids. Okay, so familiar with those. Then as far as use, the big use is to store energy, just like carbohydrates. They store energy. Sorry, my handwriting's going nuts here. But they can also be used to make up the cell membrane and as waterproofing protective layer. Okay, and then the big examples you need to know, well, besides the phospholipid and the triglyceride, you don't need to know specific examples of those, except that they are known as fats, oils, really bad handwriting, I think. Alright, fats, oils, membrane, right, membrane, and last but not least, cholesterol. I'm going to write him down here because I'm having trouble with the pen. Sorry guys, but there we go. Make sure that you fill out the online questions, and we'll see you tomorrow to talk about the video.